Today, what we're going to look at is what happens when the impulse makes it all the way to the synaptic terminals. Because if there's a neuron attached or really near this neuron here, that electrical impulse is made to send a message to the next neuron. How does that message get from one neuron to the next? Because the truth is they're not actually touching each other. There's a little microscopic space between this neuron and the next neuron. We call it the synapse, and it's a really important part of neuroscience. So this is a synaptic terminal. So imagine the impulse coming down the dendrites, the cell body, the axon, and now all the way to the synaptic terminal. When that depolarization wave, when that action potential, when that impulse gets to the end of the neuron, the synaptic terminal, it is not touching another neuron. There's a little space. So the electrical charge can't continue to the next neuron. What does continue is a chemical that gets released from this neuron to communicate with the next neuron. We call that a neurotransmitter. Has anybody heard of that term before, neurotransmitter? Will, yep. Okay, good. You may have heard that term before if you've ever learned anything about drugs of abuse or medication that affect the way that we think or the way that we perceive reality. They all work at the level of the neurotransmitter. Jason, yeah? And this becomes a really important part of neuroscience, neuroanatomy, studying the nervous system or the brain and how it works. Because if you want to control how the nervous system sends messages, you can do it by interacting with these chemicals that get released at the end of the synaptic terminal. If you know what this neuron does, what its purpose is, or let's say this neuron attached to this neuron, if you know what will happen if this neuron gets stimulated, and you want to make sure it gets stimulated, you'll find out what kind of neurotransmitter gets released from this neuron and that neuron, and you may put it in somebody's bloodstream in order to stimulate that action. Or if you know what happens when this neuron stimulates that neuron in the brain, and I'm being very, very kind of general because we look at the brain as like many cells are communicating at the same time, but let's just try to simplify it to one cell at a time. If you know what this neuron does to this neuron, you can control it by controlling the neurotransmitter. So these neurotransmitters are incredibly powerful. Most of the drugs of abuse, most drugs, medicines, psychoactive chemicals that affect how we perceive reality, how we perceive sensation, how we feel, how we react, they all interact at the level of the neurotransmitter. They either mimic these neurotransmitters, which means they basically do the same thing as these neurotransmitters, making it basically like telling this neuron to fire without that neuron telling it to. Sometimes they block the action of the neurotransmitters. So even though this neuron is trying to tell this neuron to send a message, we're saying, no, we don't want that message to get sent. So you can block the action of the neurotransmitters. You can cause the neurotransmitters to hang around longer, so that neuron gets stimulated more than it would normally. Or maybe you want to stimulate this neuron less. Maybe somebody has an anxiety disorder and certain neurons in the brain are overstimulated. Or somebody has a hard time focusing, so certain parts of the brain are understimulated. We can put chemicals into the body that control the action of these neurons, control whether they are able to be told to send an action potential or not. And so how does this work? Well, as the neural impulse travels down that synaptic terminal, it gets to the very end. We call this an axon bud. When it gets to the axon bud or the axon terminal, they have lots of different names. The electrical charge causes these little sacs to move close to the surface of that synaptic terminal. Call them the synaptic vesicles. You don't have to remember this stuff. Then that depolarization causes these gates to open to flood in the mineral calcium. Calcium has a positive charge. It actually has two positive charges. It's very positive. And it causes these vesicles to attach to the lining of that synaptic terminal and release their chemicals into the synapse, into that opening. Those neurotransmitters will then bind to these receptors. If they bind to the receptors, they bind to the receptors, they will then cause sodium to flood into that next neuron. Well, doesn't the inflowing of sodium cause depolarization? Remember all of that positive sodium going into the neuron? Doesn't it cause a cell to become positive on the inside? It depolarizes. It's exactly what an action potential is. So it starts the action potential process in that neuron, that next neuron. So we call it the initiation of action potential, if enough of that neuron is stimulated with enough sodium. Let's take some notes, and we'll talk through this process a little bit more. Please write down, and we're just continuing from where we left off last time. We're still in the same unit. The synapse. The function, it releases chemicals which take the message from one neuron to another neuron, or the message from a neuron to an organ, or from the neuron to a muscle. So depending upon whether it's neuron to neuron to some type of an organ, or neuron to some muscle in the body, neurons can tell those three different types of tissue what to do. This is accomplished. This is what I want you guys to remember and memorize. This is accomplished by calcium ions entering the axon that has the original impulse, then causing that axon to release neurotransmitters into the synapse. Now this happens really fast, and this is happening all the time right now as you're just sitting here listening. It's happening constantly in your brain. It's happening to thousands upon thousands of neurons at the same time, and those thousands of neurons connect with thousands of other neurons in the brain. And it can be this like really complex web of interactions, which can make it difficult to control the actions of the brain this way. But we've gotten pretty good at it in the field of psychopharmacology. Now, the neurotransmitter is what travels across that opening, that synapse. The neurotransmitter gets released from that one neuron that's stimulated. They float down to the next neuron, or the muscle, or the organ, and then the neurotransmitter binds to little locks, and it opens up gates for sodium to flood in. There are other type of gates that are also being opened too, but we're going to try to just simplify this as much as possible. It's a little bit more complex than what I'm explaining, but I don't want to get into all the different types of receptors at this point in the neuron because it can get a little bit confusing. And it's really not super important to us at this level. If you end up going into college and studying psychopharmacology, studying neuroanatomy, you will spend lots of time memorizing, understanding, all these different types of receptors and how they can be manipulated and to what end. This was actually the field of study that I researched when I was in college. So I spent a lot of time learning about how these different substances affect different parts of the brain, and did some very interesting research. Research on rats, which I don't really look back on positively, but unfortunately, we do have to research on animals in some regards so that we can understand how some of these things work before we start giving them to people. So that neurotransmitter then basically triggers the next neuron or organ to create or start an action potential of its own. So in essence, the neurotransmitter is the connection between this neuron's electrical activity and the next neuron's electrical activity. They don't quite touch, but they can continue the information even though they don't touch. Muscles work in a very similar way to neurons. Slightly different, and so neurons can communicate with muscles. Neurons can also communicate with some of our organs, like our heart can tell the heart to beat faster, beat slower, tell muscles of our diaphragm to increase the rate and depth of contraction so that we can breathe more or breathe less. The key is, after that neurotransmitter sends the message to the next neuron, the next structure, the neurotransmitter then gets broken down. So the neurotransmitter goes back up into the synapse, a protein grabs it, breaks it apart. So it can't keep stimulating that neuron to keep sending that message. Now, one of the ways that we treat depression is we stop those neurotransmitters from being broken down in certain parts of the brain, in certain synapses between certain neurons. We also have a mechanism in our neurons that reabsorbs some of those neurotransmitters so they can't stimulate that neuron. Other medications, newer medications for depression, actually make sure that the neurotransmitters hang out longer in the synapse in certain parts of the brain. And by doing that, you're able to stimulate certain parts of the brain more than usual. And that increases your mood. That makes you feel less depressed. Similar things we do with anxiety medications. Anxiety medications sometimes break some of those neurotransmitters down faster, depending on what part of the brain we're in. Some of them actually allow those neurotransmitters to hang out longer in the synapse too, causing the person to feel a little bit less anxiety. This is chapter 8, correct, Johnny? Okay, so I'm going to show you what I've been talking about. This is not the greatest video, but it gives you a good animation of what's taking place. I'll try to find a better video. Maybe we can watch it next week to see this a little bit much more up close. But for now, let's watch the video. I'll play it one time. Just try to pay attention to what's happening. This is a synaptic terminal. These are calcium channels. they're closed right now. This stuff up here is calcium, the green dots. The red dots or the pink dots are the neurotransmitter. Oh, I'm sorry, that's sodium. Excuse me, this is sodium. These are the receptors. They will open only to allow the sodium through if these neurotransmitters get released.
Potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
So it's going to play it again for you. Here's the action potential. Here's those calcium channels. They've just been opened because of the depolarization wave. The calcium floods in. It causes these sacs of neurotransmitters to release the neurotransmitter into the synapse. This one happens to be called acetylcholine. It's a type of neurotransmitter. Once they float to these receptors, they will cause those gates to open. See them opening, and then sodium is allowed to flood into that next neuron. And then if enough sodium floods in, you'll get depolarization of this neuron, and then it will travel all the way down in one direction from dendrite, cell body, axon, synaptic terminal. And I want to make this very clear. Sometimes the message skips the dendrite. Sometimes the neuron is attached just to the cell body like this. These are all axon terminals or synaptic terminals from other neurons. Here's another neuron, 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 here's another neuron. And they all have these synapses with these dendrites of this neuron and the cell body. So I just want to clarify that a little bit. Sometimes the impulse starts at the dendrite, sometimes the impulse begins right at the cell body. But the purpose of this is to show you that some of the messages coming from some neurons tell the next cell to go, to fire, to create an action potential. Some, however, do the opposite. They tell the next neuron to stop, to not fire, to not create an action potential. And depending on how many go messages and how many stop messages are happening at the same time will determine whether this neuron fires. So it's not just like one neuron tells one neuron to fire. It's not always the case, especially in the brain. We have neurons that have lots and lots of other neurons. We use the word in neuroscience, innervating other neurons. That means connecting to and controlling the other neurons. And you can see this electron micrograph, it's an electron microscope image of a neuron cell body and all of these other axon terminals, synaptic terminals that are attached to it. And so if we figured out what this neuron is responsible for controlling in the brain, and we put a chemical in the body that basically mimics the go message, we can control that message, that neuron better, and we can make that neuron fire more often. If we want to tell this neuron to stop sending message, then we would put some type of medicine in the body that would mimic the stop message. We call the go synapses excitatory. They excite the neuron that they're attached to. We call the stop messages inhibitory. They stop the next neuron. They inhibit it from firing. Now, we've talked in terms of neuron to neuron so far. I do want you to be aware, however, that neurons can also be attached to muscle fibers. When they're attached to when neurons are attached to muscle fibers, we call that a neuromuscular junction. Neuro for neuron, muscle, muscular for muscle cell. This would be a neuron to neuron junction. We just call it neural junction. This is a neuron to muscle, neuromuscular junction. It's the same process though. The neuron has calcium flood in. These vesicles release neurotransmitters into the muscle cell, and it stimulates that muscle cell to contract or relax. Okay, does anybody have any questions about the synapse so far, or this process of neural transmission between neuron to neuron, neuron to muscle, neuron to organ? It's incredibly crazy, huh, Will? Very, very, very crazy. I find personally the nervous system one of the more interesting parts of the body, just because of all of the small things happening and how they can be controlled at the molecular level. I've always found that very fascinating. But yeah, I agree with you. So when we've been talking about the nervous system, we've been talking about that small unit of function, that small cell that does most of the work for the nervous system. But it's not the only cells that we find in the nervous system. We usually just focused on the neuron because it's the cell in the nervous system that's doing most of the work. But there's also other cells that protect and support the neurons in the nervous system, and they're called the neuroglia. They can be found in the central nervous system, the brain and the spinal cord. They can be found in the peripheral nervous system, which are the cranial nerves, these little, we call interneurons between the central nervous system and the peripheral nervous system, and then in the spinal nerves themselves. So spinal nerves are the nerves that run from the spine to the body, from the brain to the body. There's little nerves in between the spinal nerves and the spinal cord nerves, which we call interneurons. And then there are the neurons of the spinal cord and the brain, which are the central nervous system. So let's look at some of these neuroglia before we finish up talking about nervous tissue. So we have some cells in the nervous system that are responsible for allowing the blood to bring oxygen to neurons and take carbon dioxide away from them. Blood vessels don't touch neurons. They don't really come all that near to neurons. They're not close enough to allow oxygen to travel into the neuron. Remember, neurons burn glucose. They need oxygen to burn glucose. They also need glucose in order to burn their energy. So they need oxygen in order to get the glucose burnt, and they need a source of glucose from the food we eat. But that blood travels through our blood vessels here. These are blood vessels, little capillaries. And there's cells in between the capillaries and the neurons. They basically keep the neurons separate from our blood flow. We call these cells astrocytes, and they create a nice little barrier between our bloodstream and neurons. Not everything can get from our blood into our brain, in our spinal cord. Not everything can get from our blood into these neurons here, which is a good thing. It protects them from a lot of bad stuff that could be flowing around in that bloodstream. So we call these cells and the way they interact with the neurons as the blood-brain barrier. There are other microglial cells in the nervous system that are responsible for keeping these neurons protected from debris, keeping them protected from foreign objects that shouldn't be anywhere near the neurons, and keeping them protected from viruses and bacteria that happen to make their way into the brain. We call those the microglial cells. There's also fluid that neurons float in the brain and in the spinal cord. That fluid is created by these nervous system cells called ependymal cells. You'll see it has cilia, and if you remember, simple ciliated cuboidal epithelium cells, some of them have the ability to release fluids, and that's what they do. They create a cerebral spinal fluid in the central nervous system for us. That fluid allows the neurons to float. It also allows sodium and potassium to float constantly around the outside of these neurons. Remember, sodium and potassium are really important for carrying impulses, messages through those neurons. There are other cells called oligodendrocytes that create the myelin sheath around the axons of neurons. There are other cells called Schwann cells that also create myelin to go around axons. These are found in the central nervous system. These are found in the peripheral nervous system. So let's take some final notes here. Neuroglia. Their function: support, nurture, and protect neurons. There are 6 types. First, oligodendrocytes. They produce myelin to cover the axons. Remember, myelin is that fatty tissue that covers some axons. Helps that impulse travel faster on the myelinated axons. The oligodendrocytes, I want you to remember, produce myelin in the central nervous system, brain and spinal cord. Let me fix this. This is bugging me. The microglia are in the nervous system to clean up debris, waste products, and pathogens. They're found in the central nervous system. I should probably fix this too while I'm at it. The ependymal cells produce and circulate fluid to surround the neurons, to provide a little bit of protection and cushion, also to allow for those minerals to be readily available for those neurons when an impulse needs to travel. We find these only in the central nervous system. In the peripheral nervous system, so in the cranial nerves, the ganglia, and the sensory nerves in the body, call them the spinal nerves, any of the myelinated axons have their myelin created by the Schwann cells. Don't know why that's, oh wait, that's because it's the name. In the central nervous system, the astrocytes, the ones that hold onto the blood vessels and the neurons, create a barrier between the blood and the brain. Call it the blood-brain barrier. And then finally, the cells that protect the cell bodies in the peripheral nervous system are called satellite cells. And so you'll notice there's a difference between the ones found in the central nervous system and the peripheral nervous system. So be aware, even though these Schwann cells produce myelin, they only produce them in the peripheral nervous system. In the central nervous system, we have a different type of cell called an oligodendrocyte. They're not even close in name. Now, once you write the word satellite cells, please draw a line right underneath it. And over on the left-hand side, you're going to write summary 1. Remember, the summary is at least 3 sentences about the notes that preceded it. The summary 1 gets written on the left-hand side, or the left-hand side, and then the summary itself gets written on the right-hand side. Now, before we finish up for the day, and before I give you time to work on that Chapter 8 practice packet, a little under 20 minutes, I just want to show you some of these cells and what they look like. So here's an image from your textbook. Here are the ependymal cells. This would be a cross-section of a spinal cord. There's a hole here that allows for all the cerebrospinal fluid to flow through the brain and all the way down the spinal cord. And they're lined, that hole is lined with these ependymal cells. Here is an astrocyte. It connects neurons to blood vessels. Here's another astrocyte connecting blood vessels to neurons. Here's that microglia cell that moves around and cleans debris, pathogens, nurtures and protects that neuron. These are the oligodendrocytes, and they create the myelin sheaths. Here's an oligodendrocyte. It creates the myelin sheath around certain axons. You can see it up here too. Here's some of these cells under a microscope. I don't know why it's out of order. This is an astrocyte up here. These are the blood vessels going through central nervous system, and then the neurons are the darker material, harder to see. Astrocytes kind of have like a star-like appearance to it. These are the oligodendrocytes with the myelin around the axons. Myelin around the axons. Here's an axon. Here's an axon with myelin around it. Axon with myelin around it attached to the oligodendrocyte. These are ependymal cells. Here's a Schwann cell, so this would be in the peripheral nervous system with myelin around an axon. Here are little satellite cells. They add some cushion and protection for the peripheral nervous system cells. And then this is just another image of astrocytes. They have a star-like shape to them.